The term biotechnology refers to the use of living organisms to modify human health and the environment. Throughout history, we have learned a great deal about the different organisms that our ancestors used so effectively. Increased technology has helped biologists study the organism's cell structure and function, and they have found ways to use these organisms even more effectively than our ancestors. However, biologists must have a basic understanding of cells and their many functions before manipulating their gene expression for our benefit. Understanding what makes up a cell and how the cell works is fundamental to all of the biological sciences. A cell is the smallest unit of life and the building block of all living organisms. Appreciating the similarities and differences between cell types within and among organisms is particularly important to the fields of cell biology and molecular biology. Molecular biology can be confusing because it is a subject that focuses on things so small you cannot even see them. So let's begin with big organisms and work down to the molecular level. Organisms are made up of organ systems such as the digestive system, pictured here. Each of these organ systems are comprised of individual organs. As shown in the diagram, organs such as the liver, the stomach, and the small and large intestines are part of the digestive system. All of these organs are made up of tissues. Tissues are a group of similar cells that perform the same functions, such as the epithelial tissue and the smooth muscle tissue pictured here. Tissues are made up of cells, and a cell is the smallest unit of life. Finally, cells contain organelles and molecules such as DNA. Let's examine a cell more closely. There are two general classifications of cells, prokaryotic cells and eukaryotic cells. Prokaryotes are single-celled organisms and include bacteria and some algae. On the other hand, eukaryotic cells make up more complex organisms like plants and animals.
Anatomy of the bone. Objective: to study the internal structure of a long bone. In humans, the skeletal system is made up of bones. The femur or thigh bone is called a long bone, having a shaft known as a diaphysis. And the two extremities are called the epiphysis. The epiphysis regions are covered by a hyaline cartilage. The outer covering of the bone is known as the periosteum. A bone consists of numerous blood vessels. In the diaphysis region next to the periosteum is the layer of compact bone. Next to it is the spongy bone. It consists of a network of thin, interconnected bony structures called trabeculae. The spaces between these are filled with soft marrow tissue. A look inside the spongy bone shows some bone cells like the osteoclast, which can absorb worn-out bone tissue. Another type of cell seen is the osteoblast, which synthesize new bone matrix. We also observe some stem cells which can divide and redivide to form numerous stem cells. These cells then lose their nucleus and get transformed into red blood corpuscles. Morphology of flower. Objective: to study the structure of a flower. In a garden, if we look at the hibiscus plant, we observe that at the apex of the shoot there is a bud, which is an immature flower. The bud looks green because it is enveloped by the green sepals which protect the tender bud. The bud is seen growing on a swollen base known as the receptacle. As the flower matures, it opens and the sepals fold backwards. The petals spread out and form the most prominent and attractive part of the flower. The sepals make the outermost whorl of the flower, which is known as the calyx. The petals constitute the second whorl, called the corolla. Petals are generally brightly colored to attract the insects which help in pollination. These two outer whorls are known as the accessory whorls. The next inner two whorls are known as the essential whorls. These are the male reproductive whorl, or the androecium, and the female reproductive whorl, or the gynoecium. The androecium is composed of stamens. Each stamen has a long filament and an anther. As the stamen matures, the anther bursts open, releasing the pollen grains. The innermost whorl is the female whorl, known as the gynoecium. It possesses a sticky stigma, on which the pollen gets attached, and a long style which leads into a swollen ovary. A section of this shows the presence of numerous ovules. If we take another example, like a mustard flower, we see that it possesses all the four whorls, that is, the calyx, corolla, androecium, and gynoecium, so it is known as a complete flower. The sepals are free and not fused, so they are polysepalous. The petals too are free, so they are termed as polypetalous. A longitudinal section shows that the position of the ovary is superior to that of the other whorls, so it is a hypogynous flower. The stamens are of different lengths. 4 are long and 2 are small. This type of stamen is called tetradynamous. A closer look at the stamen shows that an anther is basifixed to the filament. The gynoecium shows the ovary, style, and stigma. A section of the ovary shows the presence of many chambers with ovules, which are attached to the central axis, so it is termed as axile placentation. A recap shows that a flower has 4 whorls. 2 accessory whorls, calyx and corolla. The essential whorls, which take part in reproduction, are the androecium and gynoecium. A mustard flower is polysepalous, polypetalous, hypogynous, tetradynamous. The stamens are basifixed, and a section of the ovary shows axile placentation.